Thank you for the introduction. I hope you can hear me. The mic seems to be working now, and thanks for showing up to my seminar talk. What I want to talk about today is the research that my lab has been conducting over the last years since I got to Guam, focusing on the microbiomes of corals and dynamics in our nearshore reefs. And when I say microbiome, we've been mostly focusing on the coral-endosymbiotic algae of coral Symbiodiniaceae, those little dinoflagellate algae, as well as more recently the bacterial microbiome. Just to orient you, Guam is located in the Mariana Islands. On the left, you can see the Philippines. It's a 3-hour flight away. So we're in the tropical Western Pacific. And Guam, like islands in the region, okay. Okay, I can bring it maybe closer. Does that work better? Okay, cool. So I don't know about the gain. I don't want to play with it and then have feedback again. We'll see. Okay, so we're in the Western tropical Pacific, and similar to the other islands of Micronesia, Guam is surrounded by coral reefs. Let me see if I have a pointer. At the top, the picture, that's sort of like a tourism district in Tumon Bay. In front of it, you see a reef flat where you see the waves breaking. That's sort of like the extent of the reef flat. Corals are growing close to the reef margin and across the reef flat. And these corals essentially support local fisheries, tourism industry. That's really the economic pillar of Guam. What I didn't add here is they also provide crucial coastal protection. So similar to what just happened to Florida, last year we got hit by a major typhoon, and without those reefs, we would have essentially storm surge running straight into our business district. However, similar to reefs globally, our reefs are threatened by actually both coastal development, and the big one is climate change, in particular rising sea surface temperatures. And with that, I want to sort of switch to one of our study systems, Guam's reef flats that I just introduced. These reef flats are generally dominated by stacked monocropora corals. These corals you see over there, they're from these complex 3-dimensional structures, fish habitat, but they also break the waves that reach our shorelines. And among those stacked monocropora corals, the most dominant one is a cropora, or what we call a cropora pulchra, even though there's some revisions, the name might change. And the cropora species are particularly vulnerable to increasing sea surface temperatures. And you're probably all familiar with this, but really quick, increasing sea surface temperatures lead to the breakdown of the symbiosis between corals and their dinoflagellate endosymbionts, the Symbiodiniaceae, leading to a phenomenon called coral bleaching, because essentially with the endosymbionts, the pigments leave, the corals look pale, and usually that leads to corals either experiencing partial mortality or mortality. So Guam didn't experience coral bleaching until about a decade ago. So while other places around the world may have experienced episodes of coral bleaching in the 1990s or 2000s, little bleaching was documented in Guam. But starting in 2013 up to 2017, we had 4 semi-consecutive large bleaching events driven by increasing sea surface temperatures and also extreme low tides associated with ENSO events. And those extreme low tides led to aerial exposure, so exposure of corals to above the sea surface in shallow reef flats or lagoons during the hottest part of the day. So those two combined, high sea surface or high water temperatures plus the aerial exposure, led to a loss of 40 to 60% of coral cover across all of our reefs. So the picture on the right, that is a picture from one of those bleaching events. That's a cropora pulchra. You can see how the skeletons are white. A lot of those corals are already dead, and other turf algae start overgrowing them. This prompted the establishment of coral nurseries. That is not my work, but my collaborator, Laurie Vermundo, who's now also the director of the marine laboratory where I work. And the goal there was to conserve the diversity of our Stachynocophora corals, 8 species. So she collected samples from all those species, as well as all the populations that survived the bleaching events, and the idea being to propagate them for future outplant. I mentioned that that the mortality in these Stachynocophora beds, or like on our coral reefs in general, was around 40 to 60%, but the mortality, as you might expect, was not uniform. So up there on the right, you can see an aerial view of one of our reef flats, Agania Bay reef flat, on the west coast of Guam. And coral mortality was, or bleaching was relatively low, bleaching and associated mortality at the reef margin, but it was almost 100% as we're getting closer to shore. So like the bottom of the picture. And that makes some sense, right? There's water movement close to the reef margin that mitigates presumably some of the effects of the hot water temperatures closer to shore, closer to the beach. The water is relatively stagnant. It's very hot, and almost all corals die back. When I got to Guam, I started working with Laurie, who's running our coral reef restoration program. And we wanted to understand better what the mechanisms are for a resilience in some of these microhabitats, for example, and near the reef margin where water flow is high compared to closer to shore where water flow is low. We worked with a graduate student, James Pfeiffer, and he collected tissue samples in the field during a mild bleaching event from both high water flow areas, low water flow areas, and ran a concurrent experiment in our wet lab, rearing corals, copper corals in high water flow and low water flow. And we did transcriptomics, transcriptomic sequencing, so sequencing all the transcripts, all the RNA that is expressed in these different environments. I just want to highlight one of the findings. What we found is that genes that are associated generally with the stress response of corals were highly expressed even under normal ambient temperature in corals experiencing high water flow, while those were very lowly expressed in low water flow. And so the idea here is that corals on high water flow may actually be already primed to respond to stress of hot water once water temperatures increase. And this is a phenomenon generally described in the literature as gene front load. So the environment may just prime the corals to be ready to respond once sea surface temperatures increase. So I mentioned that the impacts of coral bleaching, of increasing sea surface temperatures aren't unique across microhabitats, across our reef flats. As you may expect, they're also not, those impacts are also not uniform across coral species. I was talking about the coral-endosymbiotic or other stachyne corals. Those seem to be the most impacted, like they are in other parts of the world. This graph shows bleaching and mortality as a function of time. This is sort of like a summary of Laurie Raimundo's long-term coral reef monitoring data set. And the one thing I would like to point out is you see this spike in bleaching and mortality in the acroporids. That's the 2016-2017 bleaching event. If we look at another group, agaricids, at the bottom here, Pavona decussata is smaller, or like an encrusting, low-growing coral that we find on our reef flats. There's almost no impact. And then fossiloporids that we find mixed in the staghorn and now Pavona stands seems to be somewhere in the middle. So like a weedy species that is somewhat tolerant. And what this seems to be leading to, just so like observing the reef flats over the years, is what I would call maybe a phase shift or a replacement of species. We're going from these three-dimensional staghorn coral-dominated reef flats to reef flats that are dominated by species of Pavona, low encrusting, corals that seem to be very resilient to increasing water temperatures. We haven't quantified this yet, but part of our pending EPSPO proposal is to actually look at what is the effect of those shifts on diversity in general. So just snorkeling or swimming on those reef flats after the transition, you can see that fish diversity and abundance is dropping, but there's probably all sorts of additional cryptic fauna that is also changing that we just don't know about. So that could be a whole change in ecosystem function. The other question is, what does this mean for the carbonate budgets of our reefs? So what is the calcification rates? Are they going to be different if we have different coral species? And that ultimately impacts coastal protection of those reefs. So my graduate student, Carlos Tremonte, who now moved on to a PhD in Hawaii at the University of Hawaii, he looked a little bit further into the shift from acroporids to Pavona. He established a set of transects on 2 of our reef flats, so like far shore, close to the reef crest, and further inshore, and tagged 3 species. I just listed the genus names, but it was Acropora pulchra, Bacillopora damocornis, and Pavona decussata. That I just talked about. So like this fast growing but not very hardy ground cover Acropora pulchra that forms these 3-dimensional thickets. Bacillopora that's somewhere in the middle, and then this hardy ground cover Pavona, the encrusting corals that seem to be taken over. And his goal was to track symbiont communities over an annual seasonal cycle. So see if there's actually differences in symbiont communities, if there's any sort of turnover or shifts throughout the season. These are the average temperatures that these reef flats experience throughout a year where we don't actually necessarily see strong bleaching. So up to about April, water temperatures are around 28 degrees centigrade, and then they rise steadily to 30, 31 on average throughout the summer until they drop again around November. And those bars are his 4 sampling time points. What Carlos did is he used a DNA ITS2 metabarcoding to characterize or to identify the species or plates of Symbiodiniaceae that are found in the different reef flats in those different species. So we have the Anguissa reef flat on the top. That was one of the 2 reef flats, and the Agania reef flat on the bottom. Inner would be close to shore, outer far shore. Generally, we find that Pavona decussata and Acropora pulchra harbor different plates of Platycopium Symbiodiniaceae. C40 for those interested in Acropora, and C1 or C3 in Pavona. Bacillopora mostly harbors Jurisdinium Symbiodiniaceae that are generally known to or thought of to be more heat tolerant, even though that may come with some metabolic trade-off. What is interesting, though, that I wanted to point out, and that we thought is quite interesting, is that in Acropora pulchra, actually, we see in one of the inner reef flat sites, so like in hotter water, closer to the shore, a shift or a difference. Not all corals or not all coppers there seem to be dominated by Platycopium, but they seem to be harboring Jurisdinium, and we're wondering if that is a potential acclimation or adaptation following bleaching events to this environment. The other thing that Carlos did was he quantified the cell densities of Symbiodiniaceae in the tissues of these 3 species throughout the season. And across those different reef flats. And when we started looking sort of at the trends, Acropora pulchra on the left behaves how I kind of would expect it. We have sort of like a dip in symbiont densities, so it's some mild bleaching, right? Some symbiont cells are lost throughout the hottest part of the year, but both Bacillopora and Pavona seem to be responding in almost the opposite fashion. The hotter it gets, or like during the hottest part of the year, they actually seem to be harboring more endosymbiont cells, which seems to me counterintuitive. So like anecdotally, just keeping Pavona in tanks, they seem to almost thrive at hot temperatures. So they seem to be very heat tolerant. How do we determine symbiont densities? With the EBSCO grant that Kenny mentioned earlier, we procured a flow cytometer, and the original idea was to use the flow cytometer to count cells in coral tissues. So we picked the tissue of the coral skeletons using an airbrush, and then run the tissue slurry through the flow cytometer. And the flow cytometer, we get this signature up here. If we plot red fluorescence, side scatter, or we could also use forward scatter. Red fluorescence, essentially the flow cytometer has a blue laser. If we shine a blue laser on algal cells, they fluoresce red due to their photopigments. So it becomes very clear which cells are potentially from the coral versus the symbionts. So we can count the cells, and then we just normalize our cell counts to 3D scans of the coral fragments that we took the coral tissue off to get some density across a defined area, for example, a square millimeter. That was my original intent for getting the flow cytometer. But then I had a graduate student, Colin Anthony. He worked with the flow cytometer, and he started noticing there's actually a lot more information that we may be able to glean from the symbiont cells. So in the flow cytometer, right, we have a light source on the left, a blue light. Cells pass individually one by one past the light source, and then we have a number of detectors. We can detect red and green fluorescence. Red fluorescence, as I just mentioned, should be generated by the photopigments of the Symbiodiniaceae cells. We notice that they also have some distinct green fluorescent signatures. After literature review, we concluded that it's most likely associated with some antioxidant proteins. So we think that is signature potentially for stress. There's also detector behind the light source, forward scatter. So the bigger the particle is, the less light will reach the center. Smaller particles should let more light pass. And side scatter. So if you have a very smooth particle, it probably doesn't scatter a lot of light to the sides. Rough particle should scatter more light to the sides. There has been some work a couple of years ago that showed that stress or thermally stressed Symbiodiniaceae, these electron microscopic pictures below here on the left, so like a smooth Symbiodiniaceae cell, on the right, a cell with like a rough texture. The idea there was that rough texture might scatter light. So if there's too much light reaching the photosystem, for example, if the symbiont cells are stressed by either too much light or hot waters, may rearrange or the shape of the cell may change to scatter some of the light. So Colin essentially looked at this data and derived a couple of phenotypic traits from these. So cell shape with side scatter is a proxy for cell shape. Cell size from forward scatter, photopigment fluorescence, and antioxidant fluorescence. Just so like one example, he did a survey around Guam to look at how the symbiont cells might change throughout the season. Again, this is a proper program from our reef plants. This plot, the details aren't too important, but May are symbiont cells. Oh, in orange, the orange dots represent a Symbiodiniaceae cells sampled during May, early on, when the waters are still relatively cold, and then the blue dots represent Symbiodiniaceae cells collected in August when waters are hottest. It seems to be a clear shift in sort of like these traits, in this case, side scatter, red fluorescence, forward scatter. Forward scatter seems to be getting smaller, indicating that the size, the individual cells might be larger. In general, there's a lot more variation as it gets hot. And yeah, so we are able to essentially start characterizing the Symbiodiniaceae communities using some phenotypic traits rather than just looking at cell counts and ITS2 metabarcodes to put some names on. So combining all those different techniques, we set out what again with Laura Raimundo and her restoration team. We were interested in understanding better what actually happens to corals when they're transplanted. So similar to what happens during coral reef restoration, you'll be sourcing coral fragments either from a nursery or from another healthy stand of corals and then transplant, presumably that induces some form of stress. In this example, I'm going to stick with a proper pulchra, but we also transplanted and currently working in a paper on the data for those species as well. We also transplanted a couple of variety species, including variety cylindrica. But the idea here was that we collected fragments at the luminar reef flat, and then we moved them to two sites into common gardens, the PD Marine Preserve, again on the west side of Guam. And we tracked the symbiont communities, the Symbiodiniaceae communities in a proper pulchra through time for about 15 months at the source site. So that's the top panel there. And we didn't see really much change in community composition over those 15 months. And even though there's some missing data, East A, East B, West A, West B, those are our common gardens. Seems that even following transplantation, the corals maintain the symbiont communities. There's this idea that transplantation stress may be mitigated by turnover of the Symbiodiniaceae communities, phenomenon known as symbiont shuffling. We did not, or I would argue, we did not really observe that. Using the phenotypic traits that Colin developed from this flow cytometric data, though, he also looked into how the signatures or how these traits change through time following transplantation and in the source colonies. Now, to look at the single plot, but I wanted to highlight a couple, and Colin summarized this in this cartoon below there. So cell density appears to change following transplantation. I guess that's sort of expected. When we transplant the corals, they seem to experience some stress, some bleaching. The other traits that changed were red fluorescence decreased following transplantation. So that might be an indication that photopigments, that we receive fewer photopigments in the transplanted cells and side scatter increase. So potentially the cells morphologically got a bit rougher. So like we're interpreting this as a sort of like transplantation stress response. And over time, after 15 months, cell numbers, cell densities increase again slightly. Side scatter goes back to essentially the starting state and approaches what the source colonies look like, but red fluorescence stayed lower than the comparative source colonies, even when they were sampled at the same time. And that could either be long-term stress or acclimation to the new environment. So maybe they have fewer photopigments in the new environment and fewer cells in their tissue. And so like I put this next step slide in here. So we now have different ways in which we can look at our corals. We can evaluate how they perform when we have a bleaching event, and also understand better what happens when we transplant corals from one environment to another, and potentially develop mitigation strategies to make those conditions better, identify the habitats that are amenable to restoration, versus those that might be a little bit of a lost cause. I now have, well, I brought one of my graduate students, Nico, who is in the audience here. And I have another graduate student, Nella Duenas, who's working on using respirometry to look at the metabolism, but also hopefully calcification rates of different coral symbiont combinations. So we have a proper pulchra with different symbionts, right? We have platypulping, we have jurisdinium. What does this actually mean for like the metabolism of the whole coral holobiome? And then also compare proper to Provona. So like, what do we see in terms of metabolism, maybe calcification rates between these different species? And The reason why I'm here is a collaboration with Kenny funded through Corda, and uh, we want to integrate. Um, I mischaracterize this. I think of this as like a multispectral fluorometer. Um, Essentially, a device or an instrument to derive or estimate photophysiological traits to identify heat tolerant. And I think of this like symbionts, but really coral symbiont combinations, right? We have different coral symbiont combinations that occur in our reefs. If we can use this as an additional measure to identify those that are particularly heat tolerant and those probably priorities for restoration. So while I'm not a restoration or coral restoration person, I'm working closely with our restoration folks and hope that our science, in essence, informs their restoration efforts. This, I would like to shift gears a little bit. So I've been talking about the photosymbionts. We also started looking at um, the bacterial microbiome. And the reason is, so climate change directly impacting coral photoendosymbionts, so-called bleaching. I mentioned early in the talk that one big issue in Guam and other islands that see development is the impact of how we use our land on our nearshore reefs. This is work by another graduate student of mine who's now in New Zealand pursuing a PhD. She looked at coral microbiome, bacterial microbiome diversity and community composition throughout the season. The top there on the right, you can see how in the wet season, rainfall drastically increases. And on the left, so below you can see her sampling site on one of our reef plants. On the left are the so like fecal indicator bacteria she measured during several discrete sampling time points. And as one might expect, during the wet season, when it rains a lot, coastal runoff leads to a lot of or like to an increase of fecal indicator bacteria, suggesting that there is contamination coming from the land. Now, the question was, how may that impact the corals? How does it impact their bacterial microbiomes? So Therese characterized this plot on the left. The first box is the diversity of the bacteria, the proper tissue microbiome. The middle represents the mucus of proper pulpa, and on the right, seawater. And so we're thinking of the mucus as kind of like a first line of defense for corals. So any so like pathogen bacterial challenge from the environment may get trapped in the mucus. As we expected, the bacterial microbiome diversity in the mucus is fairly similar to the diversity of the surrounding seawater, and it's also higher than the diversity of bacteria in the tissues. Therese tried to sort of try to use the structural equation model to sort of like formalize that relationship. And yes, indeed, rainfall and resulting bacterial water contamination have a significant impact on microbiome diversity. And actually, microbiome diversity of both the coral mucus, but also the tissues. The mucus more so the tissue. So in addition, she also used DNA metabarcoding approach using 16S rDNA to characterize the community composition of these microbiomes. The top, that's the tissue. The middle is the mucus. The bottom is the seawater. And arguably, the seawater, we have the few samples, or the few samples. I want to focus on the tissue and the mucus. So the tissue generally seems to be relatively stable in composition throughout the year. And each tissue sample was dominated by elucidate, which is this like skin colored or flesh colored part of the bar plots up there. Endozircomonas is the genus, and those are generally thought of as coral-endosymbionts, even though under some conditions Some people think they might also turn into pathogens, parasites. And then we also find in the darker brown shades, Symbiodiniaceae, Sympania, so like the chlamydia relative. It has been described as an endosymbiont from corals before. In humans, interestingly enough, it's my understanding lung pathogen, so pathogenicity, symbiosis, and so like different extremes along the same spectrum. One of the interesting things to us was that in the mucus, the taxonomic composition of these bacterial microbiomes during the dry season on the left, it mirrors very well what is in the tissues, but then when the wet season comes around, it drastically changes, drastically shifts how as a result of bacteria that are in the water. So this sort of like taxonomic, the view of taxonomic diversity of the bacteria. Bacteria do tend to evolve fast though, so we started getting interested in what's the underlying functional diversity. We're first thinking about metagenome sequencing. There's some challenges that we haven't worked through yet. So we have focused on the key, what we think are the key bacteria in these coral microbiomes, the Endosymbiotic or Endosymbiotic. Across reef flats, we have been sampling or isolating Endosymbiotic from tissues from a couple of different coral species, but we've also, again, been focusing on this dominant habitat-defining species, proper pulpra, to see if there's, even though we may have the same species or the same strains of Endosymbiotic across the reef flats, across different habitats, are there some shifts in the underlying metabolic potential or function of these Endosymbiotic strains? So this little device, so like a pocket-sized whole genome sequencer. Bacterial genomes are relatively simple to sequence. On the right, this is a phylogenomic tree of close to 30 isolates of Endosymbiotic from a proper pulpra, whose genomes we sequence. They're all highly similar, probably the same species, be the different strains, different isolates, however we want to think about that. But generally, phylogenetically, they're highly similar. But when we look at their genomes, this messy alignment on the right there shows that their genomes seem to be highly variable. There's lots of rearrangements. And this is work that's ongoing that one of my postdocs, a graduate student, Pablo de la Vega, are working on, trying to figure out what's the nature of these rearrangements. Are there differences in terms of genome content, or are they just rearranged? And what are the mechanisms of these rearrangements? So a lot of these differences between, or where we find blocks of genes to jump around, they seem to be associated with genomic islands. Some of them have all the components that would link them to bacteriophages. So there might be phage-induced islands. Some of them have some interesting features. Paolo identified one island that has the features that in other bacteria have been identified as interfering with essentially virion production and viruses. So there could be some health benefits to corals having these endosymbionts that could interfere essentially with viral reproduction. Okay, I want to switch to stick with bacterial microbiomes, but delve a bit further into one of our other study systems that I started looking at when I first got to Guam, and looking into, so like we have pollution and runoff, bacterial pollution, but one big issue is on a small island that development may lead to erosion and associated sedimentation. And this is a huge issue in Guam. This is a development that, in that construction site, it predates me being in Guam. It was about a decade ago. This is a housing development where excavation took place, soils weren't properly secured, started raining, and with the rain, all of the sediment washed out into, this is the bay right behind the marine lab where I work, basically settled on the coral reef and started smattering the corals. And we are still, even though it's developed now, we're still suffering the consequences of this. The other big issue with erosion and sedimentation in Guam is the formation of badlands, these areas of exposed soils, particularly in southern Guam. The cause seems to be mostly wildland arson. Hunters burn the savanna because the fresh shoots attract deer and makes hunting a lot easier. The problem is frequent burning over time. The savanna does not recover. Now you have exposed hardened soils that in the rains during the wet season soften up again and then get washed into our rivers and straight down into our nearshore reefs. And when you overlay where the badlands occur and where Guam's watersheds are found, sort of like a mirror, and show all of our watersheds, all of our rivers are down south. That's also where we find the badlands. And one particular sort of like study site in Guam that's the poster site for sedimentation for us with decades worth of work is Fuha Bay. We have the river emerging out of the jungle at the bottom of here. And well, on the pond, so like there's basically a ton of sediment being washed into that bay each year. As you may expect, sedimentation decreases highest, extremely severe near the river mouth, and then lowers to almost non-existent the further away we get from the river mouth. Close to the river, we find one coral species, further away, 84 coral species. And so sedimentation really impacts coral color, colony size, richness, and species diversity, with all of them increasing the further away we get from the river mouth, the less sedimentation the reef experiences. And what's the impact of sedimentation on corals? Generally increases energetic costs. The sediment has to be removed somehow. Increases disease susceptibility with the sediment. We may have pathogens entering the system. Photosystem or photosynthesis of photosymbionts is reduced, and larval recruitment is also reduced because coral larvae don't recruit to soft bottom sediments. Just a visual on the left, one of our study species, massive Varietes lobata, further away from the shore where it doesn't experience as much sedimentation. On the right, close to the river, you can see how the coral is smothered in sediment, and the process has essentially reduced the amount of tissue. We did some transcriptome sequencing, looked at RNA expression profiles across the gradient of both the host coral on the left and the Cradocopium C15 endosymbionts on the right. And what we found is that expression profiles cluster relatively neatly by site. So there's a break halfway through where we go from like severe to moderate sedimentation, where there seems to be a real shift in the genes that are expressed. If we look at what's up and down regulated, most of those genes are associated with energy metabolism, immune response. It might be expected, right? It's possibly to deal with sedimentation. There's immune challenges, and our field data were consistent with some experimental data published a few years ago. Another one of my graduate students, Justin Berg, he also looked at the bacterial microbiomes across the gradient. So on the left is the inner bay, higher sedimentation, outer bay on the right with lower sedimentation. In blue, the wet season, in red, the dry season. In the wet season, particularly in the inner bay, bacterial microbiome diversity increases, so there seems to be an influx of new species that added to the bacterial microbiomes of the Varietes corals. Overall, bacterial diversity, microbiome diversity is higher in the inner bay compared to the outer bay. The spike you see in the outer bay, that is following essentially a typhoon that passed through and led to a lot more sedimentation than usual. I just want to spend a couple minutes just looking at the microbiome composition as well. Left the inner bay, right the outer bay, dry and wet season. Again, we find Endozoomonaceae dominant. This is not Endozoomonas though. This is Endozoomonas. We haven't been able to, we've been really keen to, but we haven't been able to culture it. We would like to know what their genomes look like. In the inner bay, we find spirochetes abundant compared to the outer bay. We just look through the data. That's kind of interesting, right? There's a lot of diseases associated with them. When I think about our freshwater systems, I immediately think leptospirosis. And we also find actinomycetes to be apparently rather dominant in the inner bay. Actinomycetes have been described from coral bacterial microbiomes, but they're also really abundant in soils. So the question is, are these maybe of terrestrial origin? This is a signal of terrestrial origin. Did some statistics to look at which groups are significantly overrepresented in the inner bay. Mycetes pop up, but also dinoflagellate. And the limited knowledge I have of them is they seem to be oftentimes associated with disturbed soils. And so that could be another indication that they might be of terrestrial origin. And this is where I kind of turned a little bit into a terrestrial biologist. And we got some funding from Guam Sea Grant to start actually looking at soil microbiomes in the watershed, in fluor-based watershed. So we started moving up through, through the watershed to the ridges to look at the soil microbiomes. For one, seeing if we can figure out, like, if on the inner side we can show that there is taxonomic overlap. The other idea behind this project is, though, as well, that degraded soils, there's this idea or evidence out there that they lead to the selection for antimicrobial resistance genes in bacterial communities. The idea being that bacteria in resource-limited situations start fighting over those resources. One way in which they fight is by producing antibiotics, and to produce an antibiotic resistance gene. Actinomycetes are often associated with them, which brings it back to this overrepresentation of actinomycetes in the bay. This is a collaboration with Cal State San Marcos. And one of our other goals, in addition to using metabarcoding to like get a handle of what the soil microbiome communities are, they're also interested in using or planning on using metagenome sequencing, trying to identify those integrative elements that may harbor resistance genes to identify if this is actually a problem. If we can identify them in our soils and potentially in our waterways, in our nearshore reefs, because those may pose significant ecosystem health risks. And I would go further and say potentially public health risks. And then there's this connection and the interest. So I talked earlier about how we're collaborating with our coral reef restoration teams. The work on the ridge, where we start looking at soil microbiome. We started teaming up with, this is a picture from the Guam Restoration of Watersheds Initiative. And they're very interested in working with us and starting to understand so the microbial diversity of their soils are, and how that could potentially help them adjust, modify their restoration protocols to make them more successful. And what they do in their restoration is plant trees or try to establish trees in the badlands, stabilize the soils, and ultimately reduce the amount of sedimentation and runoff that are impacting us. And with this, I would like to acknowledge my lab, and my graduate students, postdocs, and technicians, and a number of collaborators. And thank again, Kenny, for inviting me to come here, learn these new techniques. Spend some time here to get to know the Dolphin Island sea level. Funding for this work has been coming from NSF EPSCoR, CODA, and SECA. With this, I'm ready for the questions. Thank you.